Okay, I think I'll start while people walk in. So as evolutionary biologists, we're of course interested in diversity patterns, and imbalance in diversity. So here I show you a species richness map of vascular plants. We're moving to plants now. And we can see that the main diversity is in the tropics, but also we find very species-rich areas in the Mediterranean-type ecosystems of the Cape and Australia. And diversity is not only unevenly distributed in space, of course also phylogenetically it's unevenly distributed, where we find very species-rich clades with species-poor sister clades. And this suggests that at least part of the diversity on Earth is the result of evolutionary radiations. And here I define a radiation as an increase in the net diversification rate as detected on the molecular phylogeny. And if these radiations are bounded to certain geographical areas, it could also explain why some areas are species-rich and other species-poor. And this raises questions such as what triggers radiations, but also what modulates radiations. And with modulation, I mean what causes divergence between lineages during a radiation. So in this study, we hypothesized that the interaction between extrinsic opportunities and intrinsic traits may modulate radiations. So for example, when a lineage colonizes a new environmental niche, it may evolve certain adaptations to this niche, which may lead to diversification. And if this happens in a particularly rapid fashion, it may also explain the radiation. So to illustrate this, disruptive selection may be the underlying process leading to diversification and to divergence between lineages during radiation. You can imagine a population adapting to a certain climatic niche, and then by chance some individuals may colonize a new climatic niche in space, and these individuals may have to deal with slightly different climatic conditions and therefore may evolve certain adaptations to these new climatic conditions. And this may reduce gene flow between the now two different populations, leading to speciation. But of course it could also be that these morphological adaptations evolve first, allowing some individuals to colonize a new climatic niche and therefore leading to speciation. So this suggests that this process of disruptive selection and diversification may occur more frequently or more rapidly in heterogeneous environments, where there's a fast turnover of climatic niches on a small spatial scale. So we predict in this study that rates of climatic niche evolution and morphological evolution should be higher, first of all, in clades that show radiation, so that have high rates of net diversification rates, and also in heterogeneous environments where there's a large turnover of possibilities and niches. And this heterogeneous environments as a cause for radiations has been hypothesized as one of the reasons to explain the extraordinary species richness in the Cape and Australia, so the Mediterranean climate ecosystems there, which are indeed extraordinary species rich for vascular plants in particular. And species in these regions need to deal with certain environmental conditions, such as high solar radiations in these open shrublands, seasonal drought because of the typical Mediterranean climate, and this leads also to regular fires. And this has resulted in some kind of convergent evolution, where in these regions in the Cape and Australia we find these typical shrublands with highly branched plants with small sclerophilous leaves, as you can see over here. So we tested for rates of morphological and climatic evolution in the Proteaceae family. The Proteaceae thank their name to the god Proteus, who could take different shapes and forms. And as you can see in their flower morphologies, they indeed can show a spectacular variation of shapes and forms. But not only the flowers show this variation, also their leaves show a spectacular variation, where they range from needle-like leaves to leaves with revolute leaf margins, where the margins are pulled under the leaf, to leathery leaves, to dissected rainforest leaves, large leaves, to woolly curly leaves, and everything in between. And as leaves may be the main units of plants communicating with the abiotic environment, we can expect proteaceae to occur in many different climate zones and habitats. And indeed, they go from montane forests to grasslands, rainforests, woodlands, montane heaths. But the main diversity of the 1,700 species, 1,000 species at least, occur in the Mediterranean shrublands of the Cape and Australia. And in a recent study by Elisabeth Reis and colleagues, they indeed show that proteaceae in these Mediterranean shrublands show higher net diversification rates than proteaceae occurring anywhere else in the world. So this suggests that indeed proteaceae have undergone evolutionary radiations in these open Mediterranean ecosystems. So I collected functional leaf trait data for 269 species based on 10 leaves. I looked at leaf shape using geometric morphometrics with 4 landmarks, which mainly determine the length-width ratio of the leaf. I looked at degree of dissection, which is this value P divided by D. Different measures of complexity of the leaf, so the parameter divided by the area, and the number of teeth divided by the parameter. The circularity of the leaf, which is how roundish leaves are. With a high circularity, they're more roundish, and a low circularity means more elongated or elliptical or needle-like leaves. Leaf area and effective leaf area, which is a circle drawn in the leaf, and this is directly related to the boundary layer thickness of the leaf. We collected data on specific leaf area, which is the area of the leaf divided by its oven-dried weight. And this is often a measure of how sclerophilous or hardy the leaves are, and it's also related to other traits in the so-called leaf economic spectrum. And in addition to the functional trait data, we collected distribution data and climatic data to determine the climatic tolerance of the species. We also assigned them to whether they occur in open, so this mainly this Mediterranean shrubland vegetation where they show radiations, or whether they occur in closed vegetation types, mainly tropical rainforest habitats. So to determine the climatic and morphological niches of the species, we perform principal component analyses. So here for the climatic data, each dot is a species, and as you can see, the species occurring in closed vegetation occupy a different climatic niche space than the species occurring in open vegetation. Same for the leaf morphological traits. Again, we can see that the open vegetation species occupy a different morphological space than the closed vegetation species. So to have a measure of these niches, I used the species scores on the first and the second principal components, which explain the largest part of the variation in the data sets. We built a phylogenetic tree for the proteaceae based on 339 species, using 5 chloroplast markers, and we estimated divergence times in beasts using the fossil calibrations indicated with the green stars. And then we asked, so how fast do these morphological and climatic niches change through time in the different clades? So we assigned the scores of the species of the principal components to the tips of the tree, to the represented species. And then we calculated the rate of this niche evolution in BAM, in which we can model the rate of phenotypic change on the phylogeny. And this is an example of the output of BAM, where you can see that red colors indicate fast rates of change, blue colors indicate slow rates of change, or more niche conservatism or trait conservatism in this case. So to have an idea of how fast these niches change through time, we defined clades, so open vegetation clades which show radiations, and closed vegetation clades which don't show these radiations, so indicated with O1 to 3 and C1 to 3. And then we can look at how the rates change through time. So here for the proteaceae as a whole, we see they're about 100 million years old, and these rates increase towards the present. This is as an example from the first principal component for climate. And then we can see that this signal is mainly driven by our radiating open vegetation clades, which all show an increase in rates towards the present, whereas our close vegetation plates don't really show this increase. And well, one of the reasons for this could be that, well, towards the present, so the last 20 million years, there has been some climate change and the onset of the Mediterranean type climate in the Cape in Australia, which may have triggered an expansion of this open habitat, allowing for the opportunity for colonizing new climatic niches. So we can also extract from the dam output the tip rates, so the rates of evolution at the tips of the trees. And then we can answer the question whether species that show high rates of climatic niche evolution also show high rates of morphological evolution or disparification. And indeed, with PGLS regression, we found a strong correlation. And we can also see, indeed, that species occurring in open vegetation have much higher rates, both of climatic niche evolution and morphological evolution. And again, the close vegetation species that don't radiate also show low rates of morphological evolution and climatic niche evolution. So coming back to our predictions, rates of climatic niche and morphological evolution are higher during radiations, as was shown that these open vegetation plates have undergone radiations, increased rates of diversification. And we hypothesized that as well, this is as well the case in heterogeneous environments, although we don't have a direct measure of that. But we hypothesized that these open shrublands have a higher heterogeneity, especially in climatic extremes, whereas in closed forest vegetation, these climatic extremes may be buffered by the forest cover. So especially on a small spatial scale, there might be more turnover in open habitats of climatic niches than in closed forest habitats. So I conclude that lineages which show high rates of diversification seem to also show high rates of climatic niche evolution as well as disparification. And this suggests that this interaction may modulate the Proteaceae radiations in these Mediterranean-type ecosystems. But to really fully understand this, we would need more population-level studies to really see if this ecological speciation indeed leads to reproductive isolation and ultimately speciation. So with this, I would like to thank all the people that helped me in the field in Australia and the Cape and helped collect leaf data for me, and the Swiss National Science Foundation for funding and collaboration. Thank you.
We have 1 minute for a question. Anybody would like to ask Renske?